I'm going to start with introduction. Chromatography is a vital analytical chemistry technique used to separate, identify, and quantify components within a mixture originating from early 20th century methods for separating colors compounds. It now encompasses various types, including gas chromatography (GC), liquid chromatography (LC), thin layer chromatography (TLC), and paper chromatography. The process involves passing a mixture through a stationary phase with a mobile phase, causing different components to move at varying rates due to their distinct interaction with the stationary phase. Advances in chromatography continue to enhance its precision, making it indispensable in modern laboratories.
In chromatography, we can distinguish the method into two types, which is planar chromatography and column chromatography. Planar chromatography is a method of separation where the stationary phase is supported or coated in paper pores or on a flat surface. Examples such as paper chromatography and thin layer chromatography or TLC. This method uses paper chromatography to separate a sample mixture into its individual components by hanging the paper and dip into a solvent at the bottom of the paper strip. Different with the column chromatography, which the mixture separates on the stationary phase, which is contained in a thin tube and is pushed through by gravity or pressure from the mobile phase. Example of column chromatography such as HTLC chromatography and ion exchange chromatography. The stationary phase is in a solid phase while the mobile phase is a liquid. But for today, we will only focusing on discussing about the planar type of chromatography by using the TLC chromatography as it is the most stable method to use on fruit. For the idea on how we are going to separate the component of fruit juice by using TLC, first, the fruit juice is first deproteinized by mixing it with ethanol and centrifuging to remove any proteins, and the supernatant containing the sugars is then spotted onto a TLC plate along with standard sugar solution. The plate is placed in a chamber containing a solvent mixture which acts as the mobile phase, and after the solvent front reaches the top of the plate, the sugars are visualized by spraying the plate with a locating reagent and heating. The distance each sugar travels up the plate, expressed as the RF value, depends on its polarity.
Now, we will explore two fundamental concepts, the stationary phase and the mobile phase, which are crucial for the separation process in chromatography. The stationary phase is the static part of the chromatography system. It consists of porous solid or is coated with a liquid stationary phase that remains fixed in place, either in a column or on a planar surface. Its primary function is to absorb the components being separated as solvents and analytes travel through it. In contrast, the mobile phase is the moving part of the chromatography system, also known as the eluting fluid. It traverses over or through the stationary phase, carrying with it the mixture of analytes. The mobile phase can be gas, liquid, or supercritical fluid, depending on the type of chromatography being performed. The key to chromatography lies in the interaction between the stationary phase and mobile phase. Components in the mixture interact differently with the stationary phase, resulting in varying rates of movement through it. This differential interaction causes the separation of the components. Now, let's consider an example of column chromatography. In this method, the stationary phase is a packed column filled with a solid or solid coated with a liquid. The mobile phase is a liquid solvent. As the mobile phase flows through the column, it carries the analyte mixture. Components in the mixture are separated based on their interaction with the stationary phase. In conclusion, the chromatography relies heavily on the interplay between the stationary and mobile phase. Understanding these phases and their roles is essential for achieving effective separation of components in the mixture. This technique has a widespread application in various scientific fields, from analytical chemistry to biotechnology.
In this slide, I am going to talk about objective of this experiment. The objective is to separate, identify, and analyze the different pigments and components present in fruit juice.
Hi everyone! So today I'm going to explain about procedure of chromatography of fruit juice experiment using TLC paper. Thin layer chromatography TLC is a technique used to separate and analyze mixtures. Here's our procedure for chromatography of fruit juice using TLC paper. But first, ensure all materials are clean and dry to prevent contamination. Handle the TLC plates carefully to avoid damaging the silica gel surface, and repeat the process with different solvent systems if necessary for better separation. Now, I'm going to introduce to you guys the materials needed. TLC plates, which are silica gel or other suitable material, developing chamber. You can use glass jar with a lid or tank, solvent systems, for example ethanol or water mixture, capillary tubes or pipettes, fruit juice sample, pencil for marking TLC plates, and lastly, UV lamp or iodine chamber. It is optional for visualization process. Now, I'm going to explain the procedure. Step 1. Preparing the TLC plate. Take a TLC plate and draw a horizontal line about 1 cm from the bottom using a pencil. Make sure the line is light but visible. Label the origin, start of the line lightly with a pencil. Step 2. Applying the sample. Using a capillary tube or pipette, apply a small spot of fruit juice extract onto the origin line. Allow the spot to dry completely. Step 3. Developing the TLC plate. Pour a small amount of the solvent systems, for example, ethanol and water mixture, into the developing chamber, ensuring it is shallow enough to do not touch the origin line on the TLC plate when placed inside. And place the TLC plate vertically onto the developing chamber and seal the chamber with a lid to saturate the atmosphere with the solvent vapors. Step 4. Visualizing the separation. Allow the solvent to ascend the TLC plate by capillary action until it almost reaches the top. Remove the TLC plate from the chamber and mark the solvent front with a pencil line immediately. Step 5. Visualization technique. This step is optional. If using a UV lamp, observe any fluorescent spots on the TLC plate. Alternatively, place the TLC plate in an iodine chamber to visualize spots that react with iodine vapors. Step 6. Calculating the RF values. Measure the distance from the origin to each spot, distance that traveled by the compound and the distance that traveled by the solvent front, and calculate the RF value, retention factor, for each spot using the formula. Step 7. Recording results. Record the RF values, spot colors, and any visual observations in a table. Step 8. Interpreting results. Compare the RF values obtained from the fruit juice extract with the known standards or literature values to identify the components present in the fruit juice. And lastly, clean up. Dispose of used TLC plates and solvent appropriately according to laboratory guidelines. This procedure allows you to separate the components of fruit juice based on their affinity for the solvent systems used and visualize them for analysis. Adjustments in solvent polarity and concentration can optimize separation for specific compounds if interest.
Hi, my name is NUWAIRAH, and I will be explaining about the example of results that will be obtained from the chromatography. But before that, let me first explain about the components that will be revealed from the chromatography process. So the first one is sugar. Sugar mostly contributes to the sweetness of the juice, as it is important for both flavor-wise and nutritional content of the fruit. Examples of sugar are such as glucose, fructose, and maltose. The second one is acids. Acids such as citric acid, malic acid, or tartaric acid are typical acids that are present in fruit juices. These acids is used to distinguish their concentration, which adds to the acidity of the juice. Next, pigment will be shown on the chromatography paper. Pigment will contribute to the color as well as the nutritional value of the fruit juice. There are 3 types of pigment. The first one, anthocyanins. It is responsible for red, purple, or blue color in fruits like berries or grapes. The second one is carotenoids. It contributes to the orange or yellow color in oranges or carrots. So the third one is chlorophyll. Chlorophyll is a green pigment mostly visible in vegetables such as spinach and green apples. Last but not least, we have amino acids. Amino acids are not as prominent as sugar and acid, but it is still essential for various biological functions, and it also contributes to the overall nutritional profile of the juice. Examples of amino acids are such as alanine, serine, and aspartic acid. So these are the example of results that are obtained from the thin layer chromatography. The first one you have here is dragon fruit extract, while the second one is a fruit called Carbalia cangerana. This Carbalia cangerana fruit is a type of berry that are commonly found in Brazil. As you can see on the top part, is an illustration of the movement of solvent during a chromatography process. The solvent will slowly ascend through the TLC plate to the top. As for calculating the RF value, it is calculated by the formula RF equals to A over B. A, which stands for the distance traveled by the concentration, while B, which stands for the distance traveled by the solvent. In conclusion, thin layer chromatography, or what we call as TLC, has been proven to be a valuable technique for analyzing fruit juices due to its ability to separate and identify various components present in a sample. Through TLC, the presence of components in a sample can be effectively detected and quantified, hence proving insight into quality and composition of fruit juices. This method not only aids in ensuring product quality but also supports research aimed to detect potential contaminants in fruit juices sample.